This is Biology 2, Chapter 18 on Bacteria and Viruses. In Part 2 of the video lecture, we will be discussing viruses and prions, as well as how the body gets infected. So, a virus. When we talk about our classification schemes and our kingdoms and domains, there is no viral kingdom. Technically, a virus is not a living organism. A virus is defined as a non-living strand of genetic material that's in a protein coat. Now, viruses aren't living, okay? They do not have all of the requirements of the characteristics of life. They are not made of one or more cells, etc. Viruses have no organelles. They are literally just genetic material wrapped up in a protein coat. And viruses cannot make proteins, they cannot move, and they cannot replicate on their own. That's why they are considered non-living. Viruses are very, very tiny. Millions of them can fit inside a cell. So the size of viruses are anywhere between 5 to 300 nanometers. And a nanometer is 1/1000th of a meter. And this is why so many virus particles can fit inside an individual cell. So if viruses aren't living, then where did they come from? More than likely, viruses came from parts of cells. The genetic material of viruses is similar to cellular genes, and that's why biologists think that they are parts of cells, because they do have some things in common with cells, but they do not act or behave like a cell. The structure of a virus. The outside protein layer is called the capsid. And inside the capsid, we have the genetic material. Viruses have either DNA or RNA. They do not have both, which is another reason why they are considered a non-living particle. Different types of viruses have many different shapes, but they all have the capsid and the genetic material inside. So how does the body get infected by a virus? So a virus has to enter a host cell. Now, on the diagram here at the bottom, you see an influenza A virus. All of those goofy little red and purple kind of knobbly things that are on the outside of the virus. These are, you can think of them almost like a set of keys. And what a virus does is it tricks the plasma membrane into opening the security gate. So Peter plasma membrane, if you remember our security guard of the cell city, is at the gate, and the virus comes in to the Cells R Us factory and says, no, man, I'm cool, I work here. See, here's my ID. And so the little nubblies on the outside of the virus almost act like a fake ID or a key in order to get inside the cell. So once the virus is attached, what it does is it sneaks its genetic material into the host cell. So the capsid, the protein coat, stays on the outside of the plasma membrane. Only the viral DNA or RNA makes it into the host cell. There's a couple of different ways that a virus can reproduce itself. One very common way is what's called the lytic cycle. This is a reproductive cycle where the virus basically goes into the nucleus, hijacks the CEO, and says, give me your copy machine. And says, I'm going to make copies of my DNA, and we're going to use that to make new viruses. So the lytic cycle is tricking the cell into making viral copies of DNA or RNA for the cell. So many, many cells get built up inside, or viruses rather, get built up inside the cell, and eventually that cell will literally burst or lyse and have millions of viral particles burst forth to go to infect another cell. Now you might think, well, holy cow, if there's 1,000,000 viruses inside one cell, then why don't I just drop dead within a minute? Well, the virus needs to infect many, many, many cells in order for us to feel that effect. So there is a point of time, which is usually called the incubation period of a virus, where it's kind of building up and you're getting a lot of these viral particles building up, but you don't yet have symptoms of that virus. Only when your body is kind of overtaken by so many virus particles, then you begin to feel those effects of the virus. In the lysogenic cycle, what the virus does is it says, and it goes into the cell, goes into the nucleus, hijacks the CEO of Cells R Us and says, all right, this company is now mine. These are the new blueprints or the DNA that you're going to use, and you're going to help me do my dirty work, and you're going to build the viruses for me. That's the lysogenic cycle. So viral DNA becomes permanent DNA of the cell. So that way, when the cell reproduces, we already have viral DNA copies inside. Now, this process takes a little bit longer than the lytic cycle does. Lysogenic cycle viruses are sometimes called sleeping viruses because they take so very long for the symptoms to appear. HIV, for example, is a lysogenic virus. A person can become infected with HIV but not be noticed that or diagnosed until a lot later. And HIV is a rather new category of viruses that have been identified that are called retroviruses. Retro means backwards, and the reason why retroviruses have to work backwards is because retroviruses have RNA instead of DNA for genetic material. Now, if you remember from Biology 1, when we talked about transcription or how RNA is made from DNA. Now, a retrovirus has to work that backwards. Retroviruses are very complex. They are probably, as far as antiviral drugs, the worst type of virus to develop an antiviral for because they are so very complex. Prions are another newly discovered kind of non-living particle. Prions are a protein that actually causes infections or disease. An example of this would be mad cow disease. This was a viral infection that occurred in the cattle industry a few years ago, mostly over in Europe. But what they were doing, which at least in the United States, this is checked after by the Food and Drug Administration, they were actually feeding cows ground up bits of old and sick cows that had died into their food. And so prions can survive that process. And so they're eating, these cows are eating this awful protein. And so it was a brain, it infected the brain and neurological functions. So the cows would, appear as if they were going crazy. And so the prion infection was the cause for that. So a prion causes normal proteins to mutate. And if proteins are mutated, they don't function or behave like they should. So this has been part 2 of chapter 18 on bacteria and viruses.